So this lecture is going to be over the urinary system, and on the lecture schedule I have it as a two-part. I'm just going to do it all as one lecture. It'll be a little bit lengthy. It'll be a 2-hour lecture. You can split it up into 2-hour segments if you like. There wasn't a good split point in there midway, so I'm just going to do it all as one lecture. All right, so these are the 2 urinary lectures. It's all included in this one lecture here. Okay, so go ahead and get started. We'll start looking at the functions of the urinary system and then get into all the details on how it operates. So these kidneys, this urinary system, which is these kidneys, the ureter, and the bladder, perform quite a bit of functions for us, maintaining a lot of homeostasis throughout the body. One of those functions, blood volume. Okay, therefore maintaining blood pressure. We looked at aldosterone a little bit with blood pressure, and we're going to revisit aldosterone a little bit because that's going to be where we regulate some of the blood volume. Waste products, right? This is the kind of the main function everybody thinks of urinary system, because one of the main products we got to get rid of is urea, nitrogen waste that we need to get rid of. If this builds up too much in the body, it can destroy cells. It can be deadly. So we need to get rid of this urea. This urea is the reason why, even if you were in a nice controlled environment, you will still, temperature-wise, you will still be dehydrating. Even if you're not sweating away, you'll still be dehydrating, because we have to produce at least roughly 400 milliliters of urine a day to be able to remove this urea. So it's mandatory that we remove this urea. And that's one of the main functions of the urinary system. Electrolytes, the sodium, potassium, the protons. We're going to see protons are regulated. The pH is regulated by the urinary system. We already saw the respiratory system has some regulation in pH, and the urinary system, be it bicarbonate reabsorption, will play an effect, and we'll look at that further later on in the lecture. Right, and then osmolarity. You've seen how important that is. Blood osmolarity is regulated by the urinary system, and also hormone production, and where we saw renin. Right, so we're going to go through this, get into it, get into all those factors, but very important system. When I took this class many years ago, kidney, whatever, but now that I teach it, it's like, oh man, it's amazing how much functions and how important these little kidney structures are to us in maintaining much of the homeostasis throughout the body. All right, so again, we'll look over the gross anatomy of the urinary system. We'll look at the actual process of voiding or going to the bathroom, micturition, and then we'll get into the more cellular or more microscopic level of what's taking place in our friends the kidneys here. Okay, but simply the urinary system, we got those pair of kidneys. That's where the filtration is going to take place. That's where the urine formation takes place. And as the urine is formed, it is brought into the ureters, little yellow string-like structures that lead down into the urinary bladder, where we're going to store. The kidneys, we get filtration of the blood, we get the actual production of the urine, and then the urine is traveled to the bladder by the ureters down to the urinary bladder where it's held. And once there is enough urine built up, that's when we will void or micturition, or actually go to the bathroom. Right. And then the pathway out is the urethra, and hopefully you're near the toilet when this is going to take place. So the kidneys, where the filtration takes place. You all probably remember looking at the kidneys, looking at all the blood vessels. Since we are filtering blood, there is a large blood supply that comes in to the kidneys. Okay, some of the structures, we have the outer cortex. All right, we have the inner medulla, and we have those renal pyramids there. So you can see this little structure. We're going to look at it in just a bit. Is the nephron. There are millions of these in each kidney. There are 1.25 million of these in each kidney, and this is where the filtration is going to take place and where urine is going to be formed. We're going to form the urine, and it's all going to start to be collected, and it'll be collected into first the minor calyxes, then the major calyxes, then the renal pelvis here. And once urine reaches here, it's going to travel from the ureter down to the urinary bladder and then through the urethra. Yeah. But that's the gross anatomy. Again, we'll look at this. This is where the urine is formed, but and we'll get to actually voiding reflexes, and then we're going to get into this structure here, the nephron, where it all takes place. All right, so let's look at the control, a micturition, right, or basically simply the process of going to the bathroom, the voiding reflex, okay, going pee-pee. So the process of urination, we got several muscles here. So we have the detrusor muscle making up the wall of the urinary bladder. It's made up of smooth muscle, so it is under autonomic control. Chiefly the parasympathetic is going to cause contraction of it. We have the two sphincters. All right, we have the internal urethral sphincter. Looking here, it is smooth muscle, so under autonomic control, and external urethral sphincter. Looking here, that is under voluntary control. Okay, so these are the muscles that are going to control the process of voiding, or the process of urination. So what happens is the bladder is getting full. We're making urine, and the bladder is starting to get full, getting full, and starting to get stretch on the bladder. So we really have two kind of reflexive responses in the urinary bladder. When the bladder's filling, it's not full. We're filling it up. We don't need to go to the bathroom. So we have the guarding reflex, in which the muscles, the detrusor muscles of the wall here, are inhibited, and the parasympathetic is inhibited, and the external urethral sphincter stimulated to contract, to hold in, so urine cannot escape. Now when the bladder gets full, you drink all that water, and you've filtered out that water, and you brought it in, and the bladder's getting full. Okay, that sends stretch, that causes stretch on the walls here, and that's going to send information to the micturition center in the pons, which will activate the parasympathetic neurons to contract the detrusor muscles. So now we got the urinary bladder contracting, and that's putting pressure on the internal urethral sphincter here, pushing urine through, and it also, these sensory response causes the inhibition of the external urethral sphincter, also the somatic fibers there. So this is kind of where you're doing the pee-pee. You have, this is somatic muscle, so you kind of, this is when you're doing the pee-pee dance. If you're not near the bathroom, you're trying to control, work against what your body's saying, hey, let it go, and you're trying to control this muscle, although your body's working against, saying, hey, inhibit it so you can go to the bathroom. Okay, it's inhibited. Hopefully you're close to the bathroom, and then that pushing of from the contraction of the detrusor will push out that urine. Okay, So that is the control, that is the gross anatomy of how we go to the bathroom. Now we're going to look into the actual making, the process in which we make the urine to go to the bladder. To understand urine formation, we have to look at the microscopic anatomy of the kidney. And if you look into the kidney, what you'll find is you'll find a little bit in each kidney, a little bit over 1 million of what we call the nephron. The nephron is the functional unit of the kidney. All right, and it's basically this series of tubules. So this is a tube starting here. The nephron is this cream color structure. And basically we're going to filter the blood out here to form what we call the filtrate, a fluid, and that will travel down these tubules. All right, and eventually into what we call the collecting duct. Okay, but basically there is, since we are filtering the blood, what brings the blood into this nephron here is, hopefully this name sounds familiar, the afferent arteriole brings the blood into what's called the glomerulus here, and the efferent brings it out. But one thing I want you to make note of here is the close association with these tubules with the blood supply. There is the capillary bed here where we actually get filtration, and then the efferent arteriole brings the blood out into another capillary bed called the peritubular capillaries, and these ones flanking down here called the vasa recta. Okay, so we have this close association with the nephron. So the filtrate, what gets filtered here, is not going to be what ends up in the collecting duct. What we see is as the filtrate travels through here, right? The filtrate, many items from the filtrate can actually get reabsorbed, brought from the tubules back into the blood, so this way. And then we'll also see that even after filtration, we can bring materials from the blood into the filtrate called secretion. So what actually gets filtered at the beginning of the nephron tip is not going to be what ends up here at the collecting duct. So this is how we're going to be able to regulate. We can regulate how much is being resorbed or being secreted, and this will help regulate those homeostatic factors we're talking about, the pH, the osmolarity, the blood volume. By regulating what actually ends up here as we move through the nephron will help control those factors. So let's look at the Nephron General. There are different sections of this nephron. We have the Bowman's capsule. All right, and the Bowman's capsule is the what we get the beginning region of the nephron. This is where the nephron begins. This is where the filtration takes place from the blood. And you can see the Bowman's capsule, or the glomerular capsule, wraps around what is the glomerulus. And the glomerulus is that capillary bed that is fed by that afferent arteriole. So the afferent arteriole brings blood into the glomerulus, and at the Bowman's capsule is where we're going to get the blood filtered. The filtrate will move from the blood into the tubules here, into the Bowman's capsule. Okay, once in the Bowman's capsule, they'll travel through the proximal convoluted tubule. All right, then down into the nephron loop. And we can see this is called the descending limb of the nephron loop. And then the ascending limb, since the filtrate's moving up. And then finally into the distal tubule. And from the distal tubule into the collecting duct. What we'll see is there will be many nephrons connected to an individual collecting duct. right? So the collecting duct collects the filtrate from many nephrons, right? So multiple nephrons will feed into the collecting duct, right? But as I said, as we move through the tubules, what started here, the filtrate that we started with here, is not the same as what ends up here, because along the way we are going to reabsorb. We're going to take stuff from the filtrate and bring it back into the blood, or we can take stuff from the blood and secrete it into the nephron, right? So the composition of what's in the filtrate here is quite different than the composition of what you will find that actually ends up in the collecting duct. Okay, and by regulating what actually ends up here helps us regulate those factors of pH, blood osmolarity, and so forth. Okay, and blood volume. In the kidney, you will find two types of nephrons. All right, you will find the cortical nephrons. 85% of the nephrons are of this variety. You can see this is the cortical nephron here. And what you see is more of it is up in the, here is the kind of the dividing line between the cortex is up here and the medulla is down here. And you can see the cortical nephrons are higher in the cortex, and they have these shorter nephron loops that only dip slightly into the medulla, where the other roughly 15% of the nephrons, the justamedullary nephrons, are this variety here, where they are closer to the border, just to the medulla. All right, and you can see they have these long nephron loops that flank down. But what you see here is you can see the paratubular capillaries wrapping around the paratubular capillaries here, but for the justamedullary, we have the paratubular, the vasa recta, as you probably remember from the anatomy days. Okay, these are going to be a very important structure. We're going to spend some time at looking what's taking place here, because this is going to help us regulate some of the factors in this urine production. Okay, it's going to allow us to make concentrated urine. Okay, urine with a lot less water, more solutes. So let's begin our process. Let's get into the first part of this. Is we have to first filter the blood. Okay, and where does that take place? That takes place at the glomerular capsule, or the Bowman's capsule. All right, and you can see the Bowman's capsule, the glomerular capsule, is basically surface membrane. There are two layers to it. There is a visceral layer here and a parietal layer, and then we have the capsular space in between. This is the capsular space is where the blood is going to be filtered into to start into the nephron, into the tubules. Okay, how's this controlled? The filtration, we have the afferent arteriole bringing in blood. All right, the afferent arteriole brings the blood in to the glomerus. The glomerus is that capillary bed here that is wrapped with the cells of the Bowman's capsule, or the glomerular capsule. The visceral layer is actually going to wrap around or be on the surface of this capillary bed, the glomerus. Okay. Blood comes into the capillary bed, into the glomerus, and leaves through the afferent arteriole. Okay. And as it moves in here, there is pressure. There is hydrostatic pressure, pushing force. Okay, there's pressure on there. It's going to push on here and can push fluid out into the Bowman's capsule. All right, so the P here is hydrostatic pressure. There is hydrostatic pressure going in the opposite direction from the Bowman's capsule into the glomerulus here. So we have PBS, PBG. This is hydrostatic pressure pushing in this direction. This is hydrostatic pressure pushing in this direction. And there's also an osmotic pressure that is driving fluid back from the Bowman's capsule into the blood. All right, but look at the numbers here, normal numbers in that 60 millimeters of mercury pressure here in this direction and negative 44 combined for these two forces. So the overall, the net force is we're going to be pushing filtrate out, but the driving force is that hydrostatic pressure that is built up here. And what we're going to be able to see is we can alter this a bit. We could put more blood into the glomerulus. We can dilate the afferent arterioles, put more blood in, and we can actually filtrate more coming out of here. Okay, but again, the driving force for filtration is that hydrostatic pressure. This hydrostatic pressure, the yellow arrow here, is the driving force to put filtrate into the nephron, into the glomerular tubule. Okay, so let's look at this arrangement here. Again, this is a capillary bed, the glomerulus, and it's going to be wrapped by this visceral layer of the Bowman's capsule. Right. And so that union between the glomerulus, the capillary bed, and the parietal layer of the Bowman's capsule, we form this filtration membrane. Okay, there are three main layers. We have fenestrated capillaries. If you remember fenestrated capillaries, they're the ones with the small little pores. And that's what you see here. Okay, small pores. The pores are large enough to let, say, some smaller proteins through. Ions, all this really smaller stuff. Glucose can get through. Amino acids can get through. Bicarbonate can get through. These smaller molecules can fit through these fenestrations. But red blood cells, too big. Any cellular components are too big to fit through these pores of these fenestration. Okay, the fenestrations of the fenestrated capillaries. Right. Then there is a basement membrane that adheres the visceral layer of the Bowman's capsule, and that is this here. If you remember, we had those podocytes. The podocytes, right? Podo feet, the little podocytes were the ones that wrapped around, and basically those feet from, say this is one cell and this is another podocyte cell with their little feet, they come together like this, and they create, there are little spaces in between those feet. And in between those spaces is this little filtration slit. Okay, so to make for blood, any components of the blood to get into the Bowman's capsule, it has to travel through this filtration membrane. It has to make it through these fenestrations and then through the filtration slit. All right, so this limits what can actually get filtered. What we'll see is there will be no large things. You shouldn't have cells in your urine. Okay, only small items, again, stuff like glucose, amino acids, bicarbonate ions, all your ions like sodium and chloride and potassium, those all can get filtered out, make their way through the filtration. Okay, again, what is the driving force here is the hydrostatic pressure. There's a pushing force on here that's going to push it through, but again, it has to be small enough to make its way through this filtration membrane. Okay, so this is the first process. That hydrostatic pressure is going to force filtrate into the Bowman's capsule, into the capsular space. So here's that pressure pushing it out. We push the hydrostatic pressure, forces that fluid into this capsular space. Okay, pushes it through that filtration membrane. And we have a term. We have a glomerular filtration rate. Okay, we want to know how much filtrate, how much blood is being filtered at what rate. Okay. And we see there's roughly 7.5 liters per hour over all your nephrons in your kidneys. There is about 7.5 liters filtrated per hour. You times this by 24 hours in the day, and that would get you to 100 liters a day. See the problem here? What do we say about the filtrate? The initial filtrate that is brought into the Bowman's capsule, that is filtered into the glomerular space here. Right. What we end up at the end of the nephrons at the collecting duct is much different than what we find here. Because if all of this filtrate turned into urine, we would be peeing 180 liters a day. We would just be sitting in the bathroom all day long peeing. All right, we only make, say, 1 to 2 liters a day, when we're well hydrated. All right, so most of the filtrate that comes out here, we're going to see is going to be reabsorbed. Reabsorbed meaning that it will go from the tubule back into the blood. All right, because we basically have to reabsorb. Say if we urinate 2 liters a day, we have to reabsorb 178 liters of filtrate that is coming out here. All right, so what is actually filtered here is very much different than what actually becomes the urine at the end. Now that glomerular filtration rate can be regulated. If we can regulate the hydrostatic pressure, because that's going to determine how much filtration takes place. If we can regulate how much pressure is in that glomerulus, we can regulate the GFR. And we can do that, one of the main ways is by changing the afferent arteriole. Okay, the afferent arteriole is the one that brings blood in. If it dilates and we're pushing more blood's allowed to go in the glomerulus, and we're not changing the exit of it, there's going to be more buildup of filtration. I kind of need to do this because, remember, we have changes in blood pressure. If we have high blood pressure, we would have more pressure coming in there, and that would increase the filtration rate. So you can see if we have high blood pressure out of the body, you actually see that you produce more urine because you are filtering more. But we try to regulate this. We try not to have it go too extreme or too, and so that is part of the sympathetics. Can help control that afferent arteriole. This is an extrinsic control because it's the nervous system telling the afferent arteriole to either constrict or dilate. Right? But this can also be done by autoregulation. Okay, that afferent arteriole, the stretch on it is actually going to cause it to do a countering effect, which is intrinsic. It's intrinsic in that cells of the afferent arteriole there. So let's take a look at these things and how that can change to offset or regulate the regulation or regulate the filtration. Okay, sympathetic regulation of GFR. All right, a drop in blood pressure or exercise. Both of those can be stimuluses to activate or cause constriction of the afferent arteriole. Lower blood pressure. What's the baroreceptor reflex? Is to increase sympathetic activity. Okay, what do we want to do? That'll increase cardiac output, but it'll also increase vasoconstriction of the afferent arterioles. Okay, GFR goes down. If GFR goes down, there's less urine production, and that will increase blood volume. What does blood volume do? It increases blood pressure. What was the initial stimulus? Low blood pressure. So the sympathetics can stimulate the vasoconstriction to limit the urine production, and therefore we retain more water in the blood and increase our blood volume, and that can help offset that blood pressure. All right. So that is how we can regulate via the sympathetic. The sympathetics activation is going to cause vasoconstriction of the arterioles. Okay. Renal autoregulation. Okay, we try to keep the regulation with changing blood pressures, going too high, going lower in blood pressures. We want to kind of keep the GFR within a certain range. We don't want high blood pressure to cause excessive urination. So your body trying to regulate it, keep it so it will have some increase in urination, but not this big drawn out amount. We try to auto-regulate it. That is what takes place with the intrinsic regulation. Okay, those cells. Okay, let's continue. Basically, this auto-regulation is the cells myogenic in that when there's more stretch on the afferent arteriole, its response is to constrict. Okay, you can see an increased blood pressure. If blood pressure increases, what's that going to do? The stretch on the afferent arteriole, it's going to stretch it more, and the cells of that afferent arteriole respond by causing constriction. Right? So that will, if we constrict it, that limits the flow, and that will slow down. With increased blood pressure at the glomerulus, there would be more filtration, but if we constrict the afferent arteriole, that will help maintain the GFR at a similar rate. And the opposite, if we have decreased blood pressure, there'll be less stretch on that afferent arteriole, and if there's less stretch, it will actually cause the afferent arteriole to dilate. Those cells will intrinsically dilate to help maintain, because if we have lower blood pressure, there's lower pressure here, there's less filtration, but if we open up the afferent arteriole, that will help maintain the pressure, the hydrostatic pressure here, and prevent us from making a large change in the GFR. Okay. So sympathetic, extrinsic control, autoregulation, intrinsic control, myogenic, and you can see the sympathetic is going to cause a decrease in GFR response to low blood pressure, where the autoregulation is there to try to maintain it so it doesn't go too high, too low with these increasing pressures. There's two main ways in which the cells of the kidney or cells of the nephron will auto-regulate. One of those is myogenic, and so this is in myo, being in the muscle. And so the smooth muscles of that afferent arteriole, they respond to either stretch or lack of stretch and will constrict or dilate. So remember, we're in the auto-regulation, we're trying to keep the GFR within a certain range. We don't want to go in too high, too low. So what happens, when changes of blood pressure take place? Systematic changes of blood pressure. So when blood pressure is high, what's going to happen? There's going to be more stretch on the arteriole, on the afferent arteriole, on those cells there. There'll be more stretch, and the response of those cells is, when there's more stretch, is to constrict. So there's more stretch, there's more blood pressure is going in. We want to reduce the blood pressure, and how do we reduce it? The afferent arteriole constricts, and there'll be less blood coming in, and there'll be less hydrostatic pressure here. On the opposite end of the spectrum, if there's low blood pressures, there's low stretch on those vessels, and there'll be lower pressure here. So how can we increase it? Those cells of the afferent arteriole will dilate. We dilate, send more blood into the glomerulus. That increases pressure, and we can maintain the filtration rate there. Okay, so that's the myogenic autoregulation. There are also cells here. There is this tubular glomerular feedback called the gesture glomerular apparatus. There are multiple cells here. We're going to see there are cells here when we get into the renin. This is where cells of the renin, there are these granular cells here. But there are also these macula densa cells that line this, this is the distal convoluted tubule, comes in close association with that afferent arteriole. Okay, there are cells there, and what they respond to is increased levels of NaCl in the tubule. If there's lots of flow of NaCl through there, higher pressure, they will release chemicals to the cells of the afferent arteriole, causing those afferent arterioles to constrict, right, to reduce the amount of pressure. There's lots of flow of NaCl across here. These macula densa cells do paracrine signaling because they're signaling to these nearby afferent arteriole cells. That's going to cause the afferent arterioles to constrict. Okay, so those are the modes of autoregulation, trying to keep the GFR. Even if we have changes in blood pressure, we can try to maintain this GFR. And how do we maintain it? Is we control the pressure here in the glomerulus. All right, so let's look at this process of reabsorption, not just of NaCl in water, but in general. But what we'll see, what we already talked about, we talked about there is 180 liters a day of filtrate. We only make 1 to 2 liters a day of urine. So what's taking place? Reabsorption. Reabsorption is taking material, taking water, taking solutes out from the tubule and bringing it back into first the interstitial fluid and back into the bloodstream. Remember, we have these tubules are all flanked by those paratubular capillaries that are picking up whatever is reabsorbed back from the nephron here. Okay, so besides just salt and water, many things get filtered here that we want to keep. Right? We need salt. We need to maintain our salt because that is going to be driving our osmosis throughout our body. So we need to maintain salt levels. We need to maintain these electrolyte levels. Also, glucose. It's absorbed here. In our day and times, we eat so much glucose, we might want to not be reabsorbed, but typically glucose, all the glucose is reabsorbed. Amino acids would get filtered out. We reabsorb amino acids. Bicarbonate, we also reabsorb. So most of what actually gets pushed out here, gets filtered, is reabsorbed back into the body, 99%. All right, gets reabsorbed back into the body. So what is coming out here at the glomerular capsule is not what actually ends up in the urine. It has changed quite drastically because we get this reabsorption. Okay, we're going to see most of the reabsorption for most materials, for water, for NaCl, for glucose, what's going to be taking place at the proximal tubule and then some at the descending limb. So the bulk of this reabsorption of materials takes place, 85%, is immediately reabsorbed back in. Okay. So for salt and water, let's see how this reabsorption takes place. for looking at the tubule. And the tubule is going to be lined with epithelial cells. Okay, and if you remember anatomy, the epithelial cells, those cells are polarized. They have a basal side. In this case, this would be the basal side over here, facing back into the body, back into the interstitial fluid and into those capillaries. There's the apical side that faces the lumen. This is where the filtrate is running through. Right, and then the two lateral sides, and usually they're tightly bound, right? Epithelial, there's very little, almost none extracellular space in between the cells, and they are cell upon cell upon cell. Right, so when that solute, we're at the proximal tubule, when the solute, or the filtrate comes out, the filtrate is pushed out, it is roughly 300 milliosmoles. So there's no osmolarity gradient. We need gradient for water to be reabsorbed, right? And so how do we make this take place? What do you see here on the apical side? You see this sodium and potassium. What do you think we need here? We also need ATP, right? Because here is our sodium potassium pump. And the sodium potassium pump is working. It's pumping sodium out, bringing potassium in. And so as we pump sodium out, there is a gradient. There is low sodium in here, higher sodium in the filtrate, and that would bring sodium in and then constantly gets pumped out. All right? What else will follow our sodium would be our water. So this is how we're getting this reabsorption. We get this 85% of the salt and the water reabsorbed here at the proximal tubule. You see what's the driving force for this is we have the sodium potassium pump working on this basal side, and that allows us to bring sodium from the lumen back into the bloodstream. So here's kind of a description of what's taking place here. As we said, the sodium, there's low sodium here. And why is there a low sodium intracellularly? Is because the sodium potassium pump is constantly pumping sodium in this direction back into the body, back into the bloodstream. Okay? And that is driven by the sodium potassium pump. As the sodium moves through this positive charge, right? It moves, causes chloride. Chloride can actually move through paracellularly between the cells, or okay? Also, as sodium moves, okay, remember sodium is the most important osmotically active solute, and if these cells are permeable to water, which they are, they have aquaporins. So as the sodium moves, what's going to follow it? It's not shown here, but our H2O is also going to follow the sodium. All right, and keep that in your back of your head, because as we move through these tubules, we can move water by moving sodium. Okay, as long as we're permeable to water, wherever sodium's going, water's going to follow it. Okay, and so this is how we're getting this reabsorption at the proximal tubule and also at the descending limb. Or at the proximal tubule and the descending limb, we're going to get 85% of what is water and sodium that is moving through the proximal tubule and the descending limb of the loop of Henle. Right, 85% of this water and sodium will be reabsorbed back into the body. Okay, so about 65% at the tubule, and then that other 20% to make up that 85% gets reabsorbed at the descending limb. Okay. One thing to note is this is constant. This is not factored in hydration. If you drink a lot of water, that's not going to affect. We're still going to reabsorb 85% of what is going through here. Okay, that leaves us with 15% left. Right, and this is going to allow us to modify reabsorption later in the nephron due to hydration level. So we'll see how we do this later on as we get to the distal convoluted tubule and the collecting ducts. So in those cells of the proximal convoluted tubule, we saw the driving force. How did we get reabsorption of water? It was because that sodium-potassium pump was constantly pumping out. All right, and keeping that sodium, moving that sodium, and allowing water to move it. What if we didn't have the sodium-potassium pump there? How could we get water to be drawn out? Okay, and we're going to need to do this later in the tubule. Right, what was at the proximal convoluted tubule? The osmolarity was 300, and the blood osmolarity is 300. There wasn't a gradient, so we had to have that pump. But how could we do this passively? Yeah, if we had an osmotic gradient. If we have an osmotic gradient, water is going to move to the area of higher osmolarity. And so that is the function of what's called the countercurrent multiplier system. Okay, this multiplier system is going to create a osmolarity gradient in the kidney here. All right, it's going to create a gradient, and then later on in the tubule, in the proximal convoluted or distal convoluted tubule, and more in the collecting ducts, we're going to be able to use this gradient to reabsorb water if we need to. Okay, and so as you move through the kidney from the cortex, we're isoosmotic up here in the cortex, but as you move deeper into the medulla, all right, this measure is milliosmoles. It's not shown here, but this is milliosmoles. So as you move through and down into the medulla, deeper into the medulla, you get this higher and higher osmolarity. So we get this osmolarity gradient compared to what's in the blood, okay, or in the nephron, even in the tubules. And as we move through here, you'll see, if you remember our collecting ducts, they move in this direction. So they start, say, up here where they collect from the nephrons and move down through the medulla in this higher gradient. Okay, so let's look at how this osmolarity gradient is made. It's basically made and maintained through what's called the countercurrent multiplier system. Okay, and there's going to be two parts to this system. We're going to first look at the tubule, what's happening in the tubule to make this gradient. Right, so in the descending limb, it's of the nephrons, of those justamedullary nephrons that dip deep into the medulla. Remember the ones with the long nephrons. Okay, we had the cortical and these short nephron loops where we had the justamedullary that had those really long nephron loops. Right, we're talking about these ones here with the vasa recta. Okay. So we're going to have this countercurrent multiplier. It's countercurrent in that we have the tubule, the fluids going in one direction from descending to ascending. Right, this is going to help make this gradient along with the vasa recta that are going to, remember those capillaries that are going to go in movement in the opposite direction. And the actions of these two processes keep and help us maintain that osmolarity gradient. All right, so let's look first. We'll look at the ascending. There's the descending and ascending limb of. Okay, this is the direction in which we're moving. Okay, there are two parts to this. There's the descending limb, where we are permeable to water, and then the ascending limb, where we are not permeable to water, but what we're going to be doing is actively pumping out solutes here. Okay, so here water can leave. In the ascending limb, water cannot leave, and we are actively pumping out solutes into the interstitium. Right? To get a higher osmolarity, we got to be putting out more and more solutes. All right, so we're going to see the actions taking place that help create this gradient. All right, so let's look at the ascending limb. Here's the cells. Here's the filtrate over here. This is what's in the tubule. Okay, and then here is the interstitial space. This is bringing the fluids back in the body. Okay. What do we see here? We see our sodium potassium pump is pumping sodium back in. Okay, what's going to happen as sodium moves down? We're going to fuel the secondary active transport. So here is, this creates this low sodium, creating our sodium gradient between the cell and what's in the filtrate. Okay, as sodium's actively being pumped out. Again, this is at the thick segment. What's that do? Now we have a gradient, high sodium out here, low sodium in the cell, and that's going to draw sodium in, and we can use that sodium gradient to fuel the movement of potassium and chloride into the cell. Okay, so this is secondary active transport here. We're using sodium gradient. What's the sodium gradient set up from? Our sodium potassium here on this side, on the basal side of this epithelial cell. Okay, as sodium is pumped in, and that brings higher potassium, and that's why we have to use secondary active transport. But as sodium's moved through, chloride is going to passively move through, okay, or use facilitated fusion there. Okay. Remember, there is no permeability to water. So since we're not permeable to water, but we're actively pumping out solutes, what are we doing to the osmolarity here? The osmolarity is increasing, right? Because solutes are allowed to leave, or being actively pushed out, but water cannot leave, and so water is not diluting this, so we're creating this osmolarity, higher osmolarity out in this interstitial fluid here. All right, so that is the one part. Here is the actively transporting. Okay. The descending limb. The descending limb is impermeable to salt. Okay, so you can see the fluid coming in at 300. And it's moving through this region. Remember, we have this gradient. Here's the osmolarity gradient as we move down through that loop. Okay, as we move through, what's happening? Water's leaving and the solutes become highly concentrated. Okay, the filtrate becomes concentrated. As it moves up, remember the ascending limb is not permeable to water, but it's going to actively transport out solutes. And so now solutes are leaving, but water is not. So this is what helps maintain this gradient through here. Okay, but we'll see water's leaving here. So water would dilute this gradient if we don't get rid of it. So that is going to be the function of the vasa recta. The vasa recta are going to come in this direction and pick up water and therefore help maintaining this osmolarity gradient. Okay, and you can see how, look at the numbers going in to the filtrate. It's 300 coming in to the descending limb, and what's leaving is we're at 100 milliosmoles. Okay, so we're losing basically 200 milliosmoles of solutes because we're actively pumping out. So you can see how we're getting this, we're maintaining or creating this osmolarity gradient. Okay, so this again, it's function of the nephron loop along with the function of the vasa recta to basically pick up the water that's leaving through the descending limb here. Okay, and the filtrate is moving in opposite direction of the blood in the vasa recta, the capillary beds, and so that is why we get the name countercurrent. One's moving this direction, vasa recta are moving in basically this direction here, in opposition of the movement of the filtrate, or what's moving the filtrate through the tubule. So this just kind of shows you number-wise how, say if there wasn't a gradient existed and we were just starting to make that gradient, how was it formed? And you can see as we move through, we actively pump out. And so as we continually do this and actively pump out, what do you see happening from here to here is we're starting to create that gradient. And if we constantly are doing this, actively pumping out the solutes, the vasa recta are picking it up, we're going to create this gradient. Okay, and what we say, what is this gradient important for? If we have an osmolarity gradient, if we're permeable to water, then water would move to an area of higher osmolarity, right? And this is going to be the driving force for reabsorption of water via ADH, antidiuretic hormone. If you remember antidiuretic hormone, what's it do? It increases the reabsorption of water at the kidneys, right? And it can do this because there is this osmolarity gradient. All right, we can passively reabsorb water. We don't have to have, okay, because of the actions here of the sodium potassium pumps here, we have this gradient. Now we can passively reabsorb water, and we're able to do that via using antidiuretic hormone. Okay, so that is why this osmolarity gradient in this countercurrent multiplication system multiplier is important. All right, it allows us, it's going to allow us to alter and reabsorb water if we need to. All right, and here's that effect of the vasa recta. Okay, as the vasa recta win, basically the net effect is there's going to be more gain of water in the vasa recta, right? Because remember, the descending limb, water is leaving. That would dilute this gradient. But since the vasa recta are coming through, they run parallel with the nephron, but they're moving in opposite direction. Their main effect is they pick up that water that is leaving the descending limb, and they can keep this gradient maintained. So it's an action of both the nephron loop, what's happening in the nephron loop, and this removal of water by the vasa recta. One other solute out there, again, they're still figuring out some of it. It's not completely solidified on everything that's taking place. There's still some action in that the deep regions seem to be very, very solute. And you can see, where does the pumping take place? It takes place here at the thick segment of the nephron loop. So that justifies why we got a osmolarity gradient here. And let me get my big head. But one of those solutes that is leaving through the collecting duct is urea. It's believed that in the deeper portions that urea is allowed to leave the collecting duct, and its accumulation here will further increase the osmolarity. Right, so this is kind of a newer shell, but basically a countercurrent multiplier plus this urea component is going to create, right, 300 up here, 1200 to 1400 osmolarity, milliosmolarity down in here. So effects of here plus this urea is believed to be what is all the force that is creating that gradient. So let's see how this operates. And why is this gradient important? We said it's going to be how ADH is increases reabsorption. Right? Chiefly it increases reabsorption at the collecting ducts. Okay, remember we have the nephrons up here. The multiple nephrons you can see feed into one collecting duct, and then the collecting ducts run through the medulla, where all the collecting ducts filtrates are going to get collected at that renal pelvis, and then we go into the ureter. Okay. This area is impermeable to water. Okay. And so if it's great we have this gradient, but if we're not permeable to water, this gradient is not going to be used. And if you remember from when we pumped out the solutes at this ascending limb, that filtrate came out at about 100 milliosmoles, very dilute filtrate. And as it travels through the collecting ducts here, these collecting ducts are impermeable to water. So as we move through here, since we're impermeable to water, it doesn't matter that there's a gradient here, because water cannot leave. And so what comes in at 100 milliosmol ultimately goes into the renal pelvis and will become part of the urine. This is when you get that very clear urine. But if you drink a ton of water, you scarf down a bunch of water, and you go to the bathroom later, what's it look like? It's very clear. There's not a lot of solutes. It's very dilute. And that is because you are well hydrated. We'll see there's very little ADH released, and you make this very dilute. This is what your urine, the osmolarity of your urine is going to be closer to, this 100. But what happens when we are dehydrated? When we're dehydrated, do we want to make this dilute urine? We're already low in water. We're dehydrated. Our osmolarity in our blood is coming up. Do we want to lose more water by making very dilute urine? No, we do not. We want to maintain or reabsorb as much water. All right, now here is where the gradient comes into play. Okay, ADH actions, remember this region here is impermeable to water. What ADH action is to do is to now make these cells that line the late distal tubule and the collecting ducts, make them permeable to water. Because if water can leave, it's going to go in what direction? It's going to go back into the body. It's going to leave the tubule because we're coming in at 100, and look at the gradient. There is, as we move further down, it's higher and higher osmolarity. Water's going to continually leave until the filtrate is basically matching that of the medulla. All right, so without ADH, would we end up down here? We were at 100. That was without ADH, because water, we were not permeable to water here. So what is the effects of ADH? In these cells that line this collecting duct in the late tubule here, they have these vesicles with these little purple aquaporins. Aquaporins are channels for water. Right? When ADH is not around, they are not out here on the membrane of the cells here. So therefore the cells are not permeable to water. But when ADH binds to its receptor, right, not shown here, but ADH will bind to its receptor, and that signaling will cause these aquaporins to be inserted on the membrane. Now we're permeable to water. And so that 100 milliosmol filtrate that comes down here, now with ADH on board, we're permeable to water, and water is going to leave due to this osmolarity gradient that the collecting duct is running through. When ADH levels go low again, these get recycled back and brought back into a vesicle. All right, and then we're impermeable to water again. So that is how ADH is causing the reabsorption. So you can see why this osmolarity gradient is important. Without it, ADH has no use. If we were, say, 300 out here, milliosmoles all the way through, all right, if ADH was on board, we would only end up with 300 milliosmoles. Basically, we would still be making fairly dilute urine. But this ADH, or this gradient here, allows us to make this very concentrated urine. This is when you'd go to the bathroom and it's very bright yellow because we have all those solutes. So you can kind of tell your hydration level a little bit. Way to tell is looking at your urine. Is it very clear? Or it means you're well hydrated. Is it very yellow? Possibility you might be a little dehydrated. And it's not a fail-safe. There are other things involved, but that's a good just general barometer looking at hydration levels, because when dehydrated, ADH is on board because we're causing reabsorption. You're going to have this very concentrated urine. If you're well hydrated, ADH is low, and you're going to have very dilute urine. We've now seen how ADH operates to cause that reabsorption of water. Now let's look at the control of release of ADH. So in cases of low water intake, dehydration, what's going to happen to plasma osmolarity? It's going to increase. So we have our blood, we're losing water but not the solutes as quickly, and therefore the osmolarity increases. If you remember ADH is released from the posterior pituitary. If you remember the posterior pituitary are those neurons starting in the hypothalamus and extending down into the posterior pituitary where they will release ADH. So you have osmoreceptors in the hypothalamus that respond to osmolarity. In this case, hey, osmolarity is high. What do we want to do? Do we want to lose water when our blood osmolarity is high, or do we want to try to bring back as much water as possible? Yeah, we want to reabsorb as much as possible. So in this case, we would want ADH to be high. We would want to release ADH, and that's what happens here. Posterior pituitary releases that ADH, and what's the ADH going to do? It's going to cause that water reabsorption. It causes aquaporins to be inserted. Now water can leave the tubule and be reabsorbed back into the bloodstream. On the opposite end, we have high water intake. Right? You scarf down a gallon of water just real quickly. What happens to your blood osmolarity? Now you're adding a lot of water into your blood plasma, and the plasma osmolarity will go down. All right, and now your osmoreceptors, hey, osmolarity is low. What's that signal? That should signal to decrease the release of ADH. Less ADH on board, those collecting ducts are not permeable to water, and that filtrate that comes in at 100 and now leaves roughly around 100. So we make that very dilute urine. More water is excreted. All right, so that is the control. The hydration levels determines basically our osmolarity levels determines our ADH release, and therefore determines what water reabsorption will take place in the nephrons. Now let's look at a term called renal plasma clearance. Many times we're trying to get rid of waste products or other products. We need to remove them from the blood. So how do we clear them? Right? And so we can look at this equation here. We can look at excretion. So to remove something, we have to ultimately excrete it in the urine to get it out. We have two processes that bring materials into the tubule, into the filtrate. Right? These will cause excretion. So whatever we filter, filtration rate, and what's secreted are all going into the filtrate, and which can ultimately be excreted. But there's stuff, the reabsorption rate. Reabsorption goes in the opposite direction of filtration and secretion and brings stuff from the tubule back in. So to figure out excretion, we can have filtration rate plus secretion rate minus what's being brought back into the body. Right. And so there are times when we want to measure. We want to measure, hey, filtration rate, or how fast in clearance, what's the actual, and so this equation, we can use this equation to basically judge what filtration rate is. If you want to look at, one way to look at how the kidneys are operating is looking how well they're filtrating. What's their rate of filtration? What's the GFR? And so we can do this using this equation. We can see that if we have a molecule in the blood that can be filtered here, but cannot be secreted and cannot be reabsorbed, then what's that leave us? Can't be secreted, the molecule can't be reabsorbed, then our excretion rate matches our filtration rate for that molecule. So one such molecule that can't be secreted or reabsorbed is inulin. So since inulin does not be reabsorbed and cannot be secreted right? Therefore, the filtration rate equals the excretion rate. And we don't want to go in and probe. We can go in and probe the little nephrons and look at, hey, what's the filtration rate? But since inulin is not secreted nor reabsorbed, we can match the excretion rate of inulin with that of the filtration rate. Okay, so we'll inject some inulin, we'll measure inulin in the blood, see what the measurement is, and then look at inulin that is excreted in the urine. And when we do this, this allows us to calculate glomerular filtration rate, what's taking place here. What is the filtration rate taking place? And what we come up with is roughly in a healthy about 125 milliliters per minute. Okay, that's a filtration rate. We're doing that every minute. You imagine that gives us our 180 liters that is coming out of our nephrons. Right? But remember that 180 liters, most of it gets reabsorbed, and our excretion is only 1 to 2 liters a day. Okay, but the filtration rate, healthy kidney, roughly around 125 milliliters per minute. Now let's look at kind of secretion. We discussed secretion as the process of moving stuff from the blood into the tubules. Okay, and this takes place not at the glomerulus capsule. It's not filtrate. So we're actually putting these in. We're taking them from the blood and putting them into the filtrate, into the tubule of the nephron. Okay. And this allows us to eliminate things more rapidly. If we can say if a drug can be filtered and also secreted, we can eliminate it quickly. So many of your drugs are removed via secretion process, and that is done by these transporters, these organic anion transporter, OATs, organic cation transporter, AUX. These here, you can see they are on the membrane that's facing the blood. They are brought in and then they are brought out into the urine. Okay, what's unique about these toxins and drugs that are getting removed out here, the amount of these in individuals varies quite readily. Okay, so if there is a drug that is removed via this OCT, okay, somebody that has more of these would secrete more into the thing and more into the lumen, and therefore they would clear this drug out of the blood more quickly. So many drugs, a lot of times people have to take measurements, blood levels of the drug to see where they stand because of this. If it is a drug that is moved out, say one of these mechanisms, it can vary kind of readily between individuals. So sometimes you have to take blood measurements to look at what is the levels of blood. You want to keep the levels within the therapeutic range. And depending on what the makeup of these, if it's a drug that's removed by one of these transporters, can vary between individuals. Okay. So now actually looking at renal blood flow. Now we want to look at a molecule that can be filtered as well as being secreted. Secretion allows us to clear things out of the blood more readily. Okay, if we can filter it and secrete it, we can get it out of the blood. If we can only filter it each time we go through, we're only going to filter a portion of that molecule. But if we're able to filter it and secrete it in, we can basically clear it all in one run through the nephron. And the molecule that this takes place is PAH. And what this allows us to do is allows us to measure what is the flow of blood through this glomerulus. Okay, because we can move it through and boom. And what that allows us to come up with is clearance is roughly 625 milliliters per minute. Right, so pH shows us in a healthy kidney roughly we're moving around 625 milliliters per minute. But what actually what's the GFR? GFR is 120. So you can see only about what moves through here is only about 1/5. 120 over 625, 1/5 of the blood that moves through the glomerulus is actually filtered out at the glomerulus. Okay, so inulin allows us to measure GFR. pH allows us to measure what is the perfusion, what is the perfusion of blood through the glomerular or the glomerulus here. Okay, and what we see is filtration only allows us to filter. So if we had a molecule that can only be filtered, like our inulin, only 1/5 of it comes out each run through here because it's not secreted with inulin. And now we'll look at the reabsorption of glucose. Or glucose will get filtered, amino acids get filtered. This is one of the other ways this takes place. But let's look specifically at glucose. We eat glucose for energy, and so we don't want to lose it all. We don't want to just excrete it all out in the urine. So we reabsorb. In normal circumstances, all the glucose that is filtered out gets reabsorbed. It's reabsorbed at that proximal convoluted tubule. And what we have here is we got, look at our friend, this is why I always harped on it, is our sodium potassium pump is going to be the drawing factor that allows this reabsorption to take place. Okay, this is basal side, so this is the body, this is where we're trying to bring glucose. This is glucose in the lumen. All right, coming down the tubule. And what do we see? We got sodium levels are moved out by the sodium potassium pump. So sodium is low here, and the concentration of sodium is higher over here. All right, by pumping this out, we keep low sodium over here, and that means more sodium's here. And now what are we doing? I told you this transport mechanism or membrane transport was going to come back in a fury with urinary, and you can see here we got sodium. All right, we have secondary active transport. We're using sodium to move glucose from low to high. As we increase glucose in here, glucose becomes high compared to, all right, the interstitial fluid here. As we keep loading up glucose, and now we can move to this other side via facilitated diffusion. Okay, but again, this is all drawn in by the movement of our sodium potassium pump, keeping that gradient so we can move. If there's no gradient, we can't use this secondary active transport. Okay, the key is also, why do we get reabsorption of all the glucose? Is we have enough transporters on the membranes here to be able to remove the glucose, be able to reabsorb it. Does anybody know any situations where glucose might be excessively high in the blood? And when we come to diabetes. Diabetes, if the glucose is excessively high in the blood, we're going to be filtering out excessive glucose because there is more in the blood, and we'll be filtering out more glucose into the nephron. And if it is extremely high, there's a possibility that glucose, those transporters, may be saturated, and once we pass this region, there's no more transporters further down the nephron, and so that glucose can make it pass and eventually get excreted into the urine. So typically we don't see glucose in the urine, but say if somebody had very non-controlled diabetes where glucose levels are extremely high, you may find glucose in the urine. All right, and that's showing this here is glucose filtered, treated as normal range. We get glucose, all of it gets filtered. Okay, there is a maximum though, and where, say if the levels were very, very high, okay, the plasma glucose levels, there may start getting excretion. And as we increase even higher and higher in plasma glucose levels with extreme non-controlled diabetes, you would find more and more glucose in the urine, called glycosuria. Okay, but typically we're in this range and we don't get excretion. All that glucose gets reabsorbed. We can even be, just mildly diabetic and we still don't get excretion. But when you start getting extreme uncontrolled diabetes and the plasma glucose levels climb higher, we start getting the excretion in the urine. All right, so now you guys have seen aldosterone before. You've seen aldosterone in that RAS system, that Renin-Angiotensin-Aldosterone system, where we looked at our blood pressure. And what did aldosterone do? It reabsorbs sodium and in turn reabsorption of water. So aldosterone can help regulate blood pressure. It also helps in the balance of sodium and potassium. Okay, because we can change the reabsorption of sodium and potassium depending on the levels of aldosterone. Well, reabsorption and secretion. Reabsorption of sodium, secretion of potassium. And we can regulate this, okay, via our aldosterone. We've already seen sodium is important in the regulation of blood volume. So if we regulate the sodium reabsorption, okay, remember at the proximal tubule, 85% of that sodium is already reabsorbed. So now we're just going to be playing with these smaller portions of sodium. We can alter the reabsorption of this other portion of sodium, and that can give us our sodium balance and also can help us regulate blood pressure. Okay, we'll see potassium is also important, so we also need to balance potassium, and aldosterone is involved in balancing this potassium. Okay, we already said 85, here the 90% of sodium and potassium are reabsorbed in the tubule, and also the descending limb of the nephron tube or nephron loop, right? And that is constant. That is not regulated. We don't change that. That just takes place. That reabsorption that takes place early on in the nephron. Okay, we can regulate the other 10%. And that regulation is going to come in the distal convoluted tubule. In the distal tubule, here's where aldosterone is going to have its actions. Right, so sodium reabsorption. Again, in the distal tubule, here you'll find those cells that line the distal convoluted tubule. So 8% of the total filtered. So using those values from before, 90%. By the time we get to the distal tubule, 90% of the sodium is already reabsorbed, and that is constant. 8% of the total filter gets reabsorbed at the distal tubule. Again, that is constant. This last 2% of the sodium that was initially filtered is where we can change the regulation. We could reabsorb or not reabsorb the sodium, and that is determined by the levels of aldosterone. Okay, if we need sodium reabsorbed, say we have low blood pressure and aldosterone gets released, that's going to act and can affect that last 2%. We can reabsorb the rest of that 2%, and then we'll get even more water reabsorption because the water is going to follow the sodium. Okay. How does this happen? How does aldosterone operate? Okay. You can see what it does is it has an effect on the sodium-potassium pumps in these cells of the distal convoluted tubule. Here's the body side. This is the interstitial fluid in the blood. This is the lumen side. But walking here, aldosterone has a cytoplasmic receptor. Okay, initiates transcription, right? And new protein channels and pumps are made. These protein channels and pumps, right, the sodium-potassium pumps, if we insert more of these sodium-potassium pumps, we're going to get more sodium to be reabsorbed. Okay, aldosterone also ramps up the activity of the existing sodium-potassium pumps. So we're getting more sodium-potassium pumps and we're ramping up the activity of them, and that causes for more sodium to be taken from the cell and brought in here. And of course, if it's taken from here, that causes more of the sodium to be reabsorbed. But how does, we also said that aldosterone regulates potassium. Because which way is potassium moving? Potassium's being taken from the interstitial fluid and brought into the cell, and so that would cause for more secretion of the potassium. Okay, so with aldosterone high, we get more sodium reabsorption, but we'll see we also get more potassium secreted or excreted in the urine because it gets secreted out here by the activities ramped up by the sodium-potassium pump. All right, so let's look at that potassium secretion. Okay, secreted in regions, so the later regions here is where we get the secretion. All right, and so if we're high in potassium, we can secrete more potassium. If we're low in potassium, then we're not going to secrete, and we're going to keep it into the blood. We're not going to bring it into the tubule. Okay, so you can see if we ate a banana, bananas are high in potassium. Say you scarf down a bunch of bananas. Okay, this secretion again matches that of the plasma levels. So if secretion or plasma levels are high, secretion of potassium is going to be higher. So if you scarf down a bunch of bananas, you're going to raise your potassium levels. That potassium level is going to also be a cue to release more aldosterone. So one way to cause secretion is to eat more potassium. Okay. So let's look at the cues that would control this aldosterone secretion. Remember we said high levels of potassium is one factor, and what else, right? Because if we have high potassium, we want it secreted, so aldosterone would be good because it causes secretion of potassium. Also, what else would cause? If we need more sodium, sodium levels, we need to re-up sodium levels. That may be another cue for bringing or releasing aldosterone. Okay. And we've kind of already seen this mechanism of sodium, low sodium causing a release when we saw RAS, but let's first look at high K levels, high potassium levels in the blood directly cause depolarization of those aldosterone secreting cells. So right? That's a humoral effect. It's the levels of potassium in the blood is going to cause those cells just to secrete aldosterone. How about low Na, low sodium? This causes an indirect stimulus. Okay, so low Na is accompanied by a fall in blood volume. Okay, it inhibits low Na, low sodium, inhibits the release of ADH. ADH brings water in. If ADH is not on board, it's not reabsorbing water, and what's happening in the blood? The blood volume is going to decrease. If blood volume decreases, what happens to blood pressure? Blood pressure decreases. And if you remember, going back to our system, remember, low blood pressure will be the stimulus to secrete renin from the kidneys. Right? And that takes place at the juxtaglomerular apparatus. We've already seen the macula densas. These help control the afferent arterioles, but now we have these, looking at these granular cells here. Okay, these granular cells respond to the stretch here at the afferent arteriole. If there's high blood pressure, there's more stretch, and we're not going to release renin from the granular cells. This is where the renin would be released from. Okay, the granular cells release the renin. When do we want renin on board? We want renin on board when blood pressure is low. So when there is a decreased stretch here, that's going to cause the granular cells to now secrete the renin. Okay, and back to that big ugly slide. Right, we're here. Na deficiency is what we saw. Low Na caused the low ADH, which in turn caused that decreased blood volume and that decreased blood pressure. And now we get the juxtaglomerular cells releasing renin to stop this path. We'll just walk through again. Angiotensinogen, which is an inactive molecule, is just secreted into the blood. Not at this point, it's just produced by the liver. It's running around in the blood. All right, it is an inactive molecule because it has to be converted to angiotensin 1 to start this process. What starts it, this is already in the blood, and now we get low blood pressure. That's the cue for renin, and renin will cleave angiotensinogen into angiotensin 1. We got that ACE enzyme, angiotensin converting enzyme. It converts angiotensin 1 to angiotensin 2, and now we're going to start having effects on the blood pressure. We get vasoconstriction at the arterioles, and what else do we get? This is where the angiotensin 2 causes that increased aldosterone. Aldosterone is going to increase sodium reabsorption, and in turn, what's going to happen is we're going to have reabsorption of, we have increased sodium, we'll have increased reabsorption of water because the water is going to follow the sodium, and we're going to increase the blood volume and therefore increase the blood pressure. Okay. Remember though that we're going to see that aldosterone is going to cause increased secretion of both potassium and, we haven't talked about it, but increased proton secretion. All right. So we'll see this effects of this in a bit when we see lower or higher levels of aldosterone. Okay, but this is where it begins from, the juxtaglomerular cells of the kidneys. Okay. So let's look at the renin, where I said low sodium. Low sodium causes low plasma concentrations, right? Low osmolarity. that goes, hey, we don't want to release ADH. All right, we don't want to reabsorb a bunch of water to even further dilute that osmolarity. But when we reduce the ADH, we reduce the water reabsorption, and what happens to our blood volume? Our blood volume goes down, and now we got the cue, low blood volume, low blood pressure. That's the cue for the release of renin. Okay. One other one we saw in blood pressure, and it's going to have its actions, natriuretic peptide released by the cells of the left atrium. All right, it has that opposite effect of aldosterone because it's going to cause excretion of salt. So high salt, natriuretic peptide, right? And Walter, if more salt's excreted, less will be brought back in, less water will follow it, and therefore there is more water filtered in the blood. Okay, what will that happen? That will lower blood volume and therefore lower blood pressure. So this is working in opposition. It's doing an opposite effect in that it's going to result in more excretion, not reabsorption. So it is simply the opposite effect of that of the aldosterone. All right, here's where it becomes fun in the relationship between sodium, potassium, and hydrogen ion. So aldosterone, there is a competing secretion of potassium and protons. So if levels of this change in the blood, also aldosterone will increase the secretion of these. So we'll see the effects on this and the effects that it will take place in the blood varying by how secretion takes place. But these are in opposition. If there's lots of potassium, they are competing for each other. If there's tons of potassium, potassium is going to win out, and less of the protons that we normally would secrete get left in the blood. So if I leave more of these in the blood, what's going to happen to my pH? It's going to go down. It's going to be acidic. All right, so let's see how this factors kind of mitigate with each other. So if I have high levels, let's go on the opposite. I have high levels, my blood is acidic. Okay, again, these are competing for secretion, potassium and my protons. If I have high levels of protons, all right, high levels of protons in the blood, there's going to be more proton secretion, which competes with the potassium. So what's going to happen to the potassium? The potassium is going to be left in the blood, and therefore potassium levels are going to rise. I'm going to get hyperkalemia. All right, what about the opposite? We already discussed high potassium levels. High potassium levels. So let me get my little eraser out. High potassium levels means now potassium's competing with the protons here, and there's going to be more potassium secreted, less hydrogen secretion. So this stays in the blood, and so in the blood the levels of proton goes up, and what happens to blood? It becomes more acidic. So hyperkalemia, if you ate a ton of bananas and socked a bunch of potassium into your blood, you're going to make your blood more acidic. Okay. Now let's look at aldosterone. So aldosterone's effects, it it has an effect on both of these ions. It has an effect on potassium, and it has an effect on protons. All right, it increases the secretion of both. All right, so what would if I had abnormally high levels of aldosterone? What might happen to my blood for these two ions? If I say I have a tumor in my adrenal gland that constantly secreting out aldosterone, what's going to be the levels of this in my blood? It was going to ramp up the secretion. Normally I have a set amount of aldosterone, so I keep this relatively within a certain range, but now aldosterone's causing, that increased aldosterone is causing massive secretion of these, and what's going to happen? I'm going to have hypokalemia, because I'm secreting lots of potassium. And what do you think pH-wise what my blood would be in this case? It would be alkaline. It would be basic because I'm doing more excretion of the protons, and therefore I'm leaving less in the blood, and now my pH is going up. And then what if I had an autoimmune disease that killed off my cells that secreted aldosterone? I'd have low levels of aldosterone. Now what would happen? Normally I have a fairly normal range of the secretion. Now I have very low levels. I'm not causing any of this, and so this will be retained and this will be retained in the blood, and now I'm going to hyperkalemia, or what's going to happen to my pH. My pH is going to be acidic. I'm going to be going down in pH. All right, because I'm retaining these, where normally I have some aldosterone, basal aldosterone, that's causing secretion of this. With these low levels, it gets retained, and same with the potassium. All right. So here's the part in the respiratory we kind of alluded to in the respiratory portion, because if you remember, respiratory can help regulate pH by regulating CO2 levels, and the urinary system can help regulate pH by regulating the levels of bicarbonate. And so here is where this is taking place. We're in the proximal tubule and also the distal tubule, but in the proximal tubule, we'll see what's taking place in that this is where we get the most reabsorption of bicarbonate. There is our bicarbonate that was filtered. Okay, it was filtered out here. It's small enough to be filtered into the filtrate. Now, there is secretion of protons. Bicarbonate doesn't get reabsorbed as bicarbonate. There is Ca, the carbonic anhydrase, and what does that do? It converts, okay, this proton combines with the bicarbonate that's filtered to make carbonic acid, and now that carbonic acid is converted to water and CO2. It is the water and the CO2 that get reabsorbed out of the lumen. Once in the cell, the carbonic anhydrase in the cell converts this to the bicarbonate, and now bicarbonate will be reabsorbed back into the bloodstream. Okay, so it doesn't get reabsorbed as bicarbonate. It has to first get converted to H2O, to water and CO2, and then it gets converted back, and boom, bicarbonate goes reabsorbed, and what happens is the proton gets secreted out. Okay. So therefore in the urine typically there is very little bicarbonate, but we can alter this depending on what the pH level is in the blood. This reabsorption of bicarbonate we can alter here at the kidneys. Okay. What would we want? Will we want more bicarbonate? Will we want more or less bicarbonate if my blood is acidic? If I have lots of protons in my blood, do I want more or less of this? I would want more bicarbonate reabsorption to buffer out those protons. I don't want my blood to be too acidic. What if I had low levels of protons? I would want less bicarbonate to buffer. So that is what's happening with these cells here at the urinary, at the proximal tubule. They can alter the amount of reabsorption of this bicarbonate depending on the acidity of the blood. All right, so under normal conditions, bicarbonate, most of it is reabsorbed. There's very little that goes into the urine. What happens when my blood is acidic? I'm going to have higher levels of this. Okay, I want more bicarbonate reabsorption. If I have higher levels of this, I'm going to be able to convert more. There will be more of this out in the lumen, and there will be more chances to make this process take place. If I do this pathway more, I'm going to reabsorb more bicarbonate. Okay, so by having acidic blood, I'm going to cause more reabsorption. Also, these cells can make bicarbonate and secrete it, so they can make bicarbonate and secrete it in, and they will do that in response to highly acidic blood. Okay, what happens with alkalosis? Alkalosis would be the opposite. There would be lower levels of protons. All right, there would be lower levels of protons out here. Lower levels of proton, that means there's less to convert to CO2 and H2O. So if there's lower amounts of this conversion, there will be lower amounts of bicarbonate reabsorption. And that's what we want to take place when there's low levels of protons. We want less of this. And so these cells will reabsorb less bicarbonate to help offset. So there's the factors. We got the CO2, right? Say we had a problem with the levels of this in the blood, we can change our CO2. Right? What happens if there's changes in CO2? We have a respiratory disorder that changes our CO2 levels and therefore changes our acidity. Here we can modify the levels of bicarbonate in the blood to offset those effects. Okay, so pretty unique organism, and it's got all these got to play in blood pressure, got to play in pH, got to play in osmolarity. These kidneys are very important little structures that regulate a lot of homeostasis that's going on in the body. This alludes back to that previous part in that their nephrons, say if we have high acidity, we can only produce urine that is roughly 4.5 pH. So what if we needed to excrete? What if our pH was extremely high and we needed to get rid of more of these? We have a buffer. We have a phosphate buffer system in that distal convoluted tubule. Right? We have buffers. We make a buffer that helps buffer it out, bring the pH back up, and allows us to keep excreting more protons. All right, so we have buffers in the blood, but we also have buffers in the urine to allow us, if need be, we can help us excrete more of these protons to help bring down the pH. All right, here is where we bring it all together. The diuretics. So many, we talked briefly on the hypertension and blood pressure, and we mentioned diuretics. Okay. Because if we have high blood pressure, what will we want to do with our blood volume? If we can regulate blood volume, we can help regulate blood pressure. And this is usually a longer term effect where the baroreceptor is very quick. This regulation of blood pressure is longer acting, and therefore, if we're able to regulate that blood volume, we're able to regulate blood pressure. Okay. The ones I really want to focus on are these sodium ones, because what causes it? If we're not reabsorbing sodium, what else won't we be reabsorbing? We won't be, the water won't be following the sodium. Okay, so you can see many of these drugs, many of these diuretic medications, what do they inhibit? They inhibit sodium reabsorption. This inhibits aldosterone and therefore inhibits sodium reabsorption. Okay. Inhibits sodium transport, So many of these diuretics, their effect is to prevent the sodium being reabsorbed, and therefore what will happen to the reabsorption of water? The reabsorption of water will also go down. And what will happen with our blood pressure? Our blood pressure will go down. So many of these hypertension drugs work at the nephron to inhibit the reabsorption of sodium, and therefore the reabsorption of water, and therefore bringing blood volume down. Okay, so this is actual mechanism to what we saw earlier when we were looking at the blood pressure. This is where these diuretics are operating. Okay, what happens when our kidneys start to go in this direction? Fortunately, this is what happens with diabetics. Your kidneys start to degrade. We get loss of nephron. We get loss of the function. The good news is that we are able to operate with about 15% of our kidney use. So we were born with an excess amount of nephrons, an excess amount of activity in the kidneys that we have to really get them destroyed before we have to take some serious actions. Okay, but what would happen with renal insufficiency? Okay, we might get tension of Na, tension of water, urea. These are all things that we want to help maintain. Okay, so we'll have some problems. The main function talked about is to get urea out. One of the main functions of the kidneys is to remove urea from the blood, okay, because this can get highly dangerous. So to do it, when our kidneys start getting down below that 15% mark, what we have to do is we have to do dialysis. We have to take the blood out and remove the stuff we need to remove, but we also have to retain in the blood, because this blood's going to be returned back into the patient. We have to retain the good stuff in the blood, right? Because our kidneys, what do? They filter blood glucose, but they bring the glucose and they bring the sodiums, they bring all those ions back in. What they're filtering out and what's getting excreted in the urine is the waste products like urea and so forth. So we want to be able to we want to remove the urea from the blood, but we want to keep, say, glucose, most of the sodium in there. And so how do we do this? via dialysis is we're going to use diffusion. The property is diffusion to maintain these levels. So what happens is we pull out blood moves this way, and there is the dialysis membrane. If you remember, the dialysis membrane is small pores. So anything small can come out. Glucose is small, urea is small, all right? Those can all move out. So how do we keep them in? What we need to do is we need to make concentration gradients. This dialysis fluid should have low amounts of urea because we want to pull urea out of the blood. But if we keep physiological levels of, say, glucose, of sodium, potassium, okay, then there is no concentration gradient, and what will take place? There won't be any net movement of glucose. So that is how we solve this problem, because we don't have the ability to reabsorb. We just have these small holes that allows molecules to move through. So if we have low urea here and there's high urea in the blood, all right, the urea is going to move into the dialysis fluid. But if we keep physiological levels of, say, sodium and potassium and glucose, then there is no concentration gradient and there's no driving force to remove that from the blood. Okay, and so that's essentially, it's a little more complex, but that's essentially what's taking place with dialysis. We're able to remove those waste products while keeping the other products. All right, and that is it for urinary. I know it's a little bit of a, we got a lot going on, a lot of molecules moving back and forth, but just get it down and we shall be good. Alrighty. Next up is digestive. Keep digestive pretty simple, so we'll be on our way there.